After brief introduction about antibodies. Here in this section we will learn about elucidation of antibody structure, or how experimental data lead towards determination of present antibody structure. Starting with finding about the class of antibody. Since the late 19th century, it has been known that antibodies reside in the blood serum. We know that serum is the component of the blood which is left behind once cells and clotting proteins have been removed from blood. However, the chemical nature of those antibodies remained a mystery until 1939. Then an experiment was performed to determine the chemical class of antibody. In this experiment, scientists immunized lab animal with ovalbumin, the major components of egg whites, and then collected anti-serum sample from the subject containing antibodies against ovalbumin, and divided into two portions. Then they subjected the first portion to the process of electrophoresis, thus measuring the amount of protein that moved different distances from the origin within the electric field. The graphical result showed the presence of 4 major protein subpopulations. The first and largest is the albumin peak, the most abundant protein in serum, with responsibility for transporting lipids through the blood. They named the other peaks globulins. The 2 smaller peaks they denoted the alpha and beta globulin peaks. The third globulin peak, gamma globulin, clearly represented a set of proteins in high concentration in the serum. Then they took second portion of that sample and treated it with the same antigen, that is ovalbumin. That is actually a multivalent antigen. That means an antigen that have ability to bind more than one antibody. Antibodies mix complex with that antigen and fall out in a process called immunoprecipitation. Now they again plot a graph to observe which peak was mainly affected. There, very little protein was lost from the albumin peak, or from the alpha and beta globulin peaks. However, immunoprecipitation resulted in a dramatic decrease in the size of the gamma globulin peak, demonstrating that the majority of their anti-ovalbumin antibodies could be classified as gamma globulins. We now know that most antibodies of the IgG class are indeed found in the gamma globulin class, and hence termed as immunoglobulins. Knowing the class of serum protein into which antibodies fall was a start, but immunochemists next needed to figure out what antibodies looked like. So other series of experiments were performed to determine the fragments of antibodies. In first set of experiment, they treated the Ig antibody, that is the simplest antibody, with a proteolytic enzyme named papain. This treatment produced 3 fragments, 2 of which were identical while the third one was different. This third fragment that was different from the other 2 spontaneously crystallized out and was therefore named as fragment crystallizable, or FC. This fragment had no antigen binding capacity, while the other 2 fragments were shown to retain the antigen binding capacity of the original antibody. These fragments were therefore named FAB, or fragments antigen binding. This experiment indicated that a single antibody molecule contained 2 antigen binding sites and a third part of the molecule that did not participate in the binding reaction but could readily form crystals, so was likely to be very similar among different antibodies. Then in another experiment, scientists treated Ig with another proteolytic enzyme, namely pepsin. This treatment resulted in the formation of single fragment, which contained 2 antigen binding sites that were still held together in a bivalent molecule. Because the molecule acted as though it contained 2 FAB fragments, but clearly had an additional component that facilitated the combination of the 2 fragments into one molecule, it was named Fab2. Pepsin digestion does not result in a recoverable FC fragment, which is apparently digested by the enzyme. This experiment somehow showed the presence of some linkage between FAB portions of the antibody, and confirms presence of dual binding sites. Then in another set of experiment, investigators reduced the whole Ig molecule using beta-mercaptuthenol that breaks disulfide bonds and alkylated the reduced product so that the disulfide bonds could not spontaneously reform. They then used a technique called gel filtration to separate and measure the size of the protein fragments generated by this reduction and alkylation. In this way, it was shown that each Ig molecule contained 2 heavy chains, with molecular weights of 50,000 Dalton and 2 light chains of molecular weight 22,000 Dalton. So until now, it was discovered that antibodies belong to the class of immunoglobulins. Second, it has 2 antigen-binding fragments and 1 crystallizable fragment that helps in receptor binding. Third, there is some linkage between Fab portions. Fourth, simplest antibody molecule comprised of 2 heavy chains and 2 light chains. Now the next challenge that scientists were facing was to determine which of the chains was implicated in antigen binding and which chains contributed to the crystallizable fragments. So to determine that, they perform another experiment, in which scientists use F, a B and F, C fragments purified from rabbit eye, GG antibodies, and immunized 2 separate goats. From these goats, they generated anti-Fab antibodies and anti-F-C antibodies, which they reacted in separate experiments with the heavy and light chains. Anti-F-AB antibodies bound to both heavy and light chains, and therefore the antigen binding site of the original rabbit eye, GG, was made up of both heavy and light chain components. However, anti-F-C antibodies bound only to the heavy chains, not to the light chains of the eye, GG molecule, demonstrating that the F-C part of the molecule was made up of heavy chains only. Now, question arises, what chemical structure make it possible? Well, we know antibodies are immunoglobulins. That means they are made up of polypeptide chains. So antibodies are made up of 4 polypeptide chains. Careful protein chemistry demonstrated that both light and heavy chains have 2 terminals. At one terminal they have amino group, so that end is called amino terminus, while the other end has a carboxyl group, and hence it is named as carboxy terminus or C terminus. This amino terminal resided in the Fab portion of the molecule, while the other carboxyl terminal is resided in the Fc portion of the antibody. So all in all, these set of experiments revealed that all antibodies share a common structure of 4 polypeptide chains, consisting of 2 light chains and 2 heavy chains. Each light chain is connected to its partner heavy chain by a disulfide bond between corresponding cysteine residues. The 2 heavy chains are also connected to one another via disulfide bonds located outside of the antigen-binding regions. Each antibody molecule has 2 antigen-binding sites called as Fab regions, and 1 crystallizable region called as Fc region. The antigen-binding sites are made up of components of both the heavy and light chains, whereas the Fc region is made of only heavy chains. Let's have a complete description of all these structural components of antibodies in our next section.